Hey folks, moving on now, we're going to start looking at some membrane protein stuff and starting to look at the actual pieces of the membrane which we could say actually do things. So let's get going with that. We talked about the fluid part, that's the lipids and their ability to move laterally, diffuse, rotationally, and rarely flip-flop. The mosaic part of the fluid mosaic model that you'll have heard about includes the kind of lipid raft component, other kinds of lipid microdomains, but primarily it's the membrane proteins that give the mosaic to the fluid mosaic model. So membrane proteins have different hydrophobicities, and so they associate with or occupy different positions in or on the surface of the membrane. So when we talk about the surface of the membrane, that can be the surface on the outside, the outer leaflet surface, or the inner leaflet surface. So depending on how a membrane how a protein associates in the membrane, affects how easily we can isolate the protein from the membrane to study it. And in fact, the ease with which we can remove membranes from proteins is the basis of how membranes were initially characterized and categorized, if you like. So we got three general categories that we can think about. Integral membrane proteins to start with, peripheral membrane proteins, and then lipid anchored proteins. Now, to be honest, I've often linked lipid-anchored and integral membranes together, and that's how I learn it, but your book separates them, so we'll keep them separate, just so there's no ambiguity. So integral membrane proteins they're said to be embedded in the lipid bilayer, and they have these extensive hydrophobic regions. Here's a monotropic protein, only half embedded into the membrane. This is a single-pass integral membrane protein, a multipass protein, and then here there's a protein which has got one transmembrane segment, and it's forming a dimer, and it's associated with a peripheral membrane protein. The peripheral membrane protein there can be associated with the phospholipid heads, or it could be associated with an intracellular domain of an integral membrane protein that has a transmembrane region. So if you think back to that activity, that first homework problem where I had you work out whether a helix was amphiphilic, one face hydrophobic, one face hydrophilic. If these two proteins are associating via these helices, then the faces of the helix which touch one another will need to be hydrophilic, but the faces of the helix which face the lipid tails, they're going to have to be, or the phospholipid and glycolipid tails, they have to be hydrophobic. And so we learned some information about the nature of a transmembrane protein by looking at whether it's amphiphilic and how the amino acids that are hydrophobic and hydrophilic kind of align down the helix. So that's one of the reasons we did that, is so you get a better understanding when we get to this point. So those are the integrals, those are the peripherals. The peripheral proteins are hydrophilic and they're associated with the surface, like we say. Our third group are the lipid anchored proteins. Now they're hydrophilic. So here's a couple of lipid anchored proteins. There's one here and there's one here. Now they're hydrophilic, so they're not embedded in the membrane in the sense that their protein component is embedded in the membrane, but they have these hydrophobic structures which are attached to the protein, which then embed into the membrane. So that could be a fatty acid, that's literally a chain of hydrocarbons, or it could be an isoprenyl structure, and there are also what are called GPI anchors, and so you see GPI anchors in the outer leaflet, and then fatty acids and isoprenyl in the inner leaflet. So there's a real quick overview. Let's take a look at just some examples here, so you've seen some stuff. Most membrane proteins are integral in nature, and they possess one or more major hydrophobic regions that have an affinity for the interior of the lipid bilayer, for the interior of the hydrophobic core. So this contributes to their assembly. We talked about self-assembly. If you've got a protein which has got a hydrophobic region, let's just look at this one in the middle here. If these coils here, if these helices here have hydrophobic faces, the self-assembly is going to be that if you put this protein with membrane lipids, the lipids are going to form a hydrophobic kind of wrapper around this hydrophobic core, not around these pieces up here, which they look disordered, but they could have some other structure as well. And so there's a self-assembly component here that emerges from the fact that there are these hydrophobic transmembrane regions. These are all integral transmembrane proteins, and as you can see, there's hydrophobic regions which allow them to embed into the membrane. These are really difficult to extract from membranes. You can't extract them with salt. You have to extract them with high concentrations of detergent. And the problem with that is that you often denature the protein, so that makes the X-ray crystallography of integral membrane proteins very, very challenging. They have to be extracted and then refolded or extracted somehow in their native state, and that's technically challenging. We talked a little bit about monotropic proteins just now. Just to recap that, a monotropic protein is a protein which is globular, largely hydrophilic, but it has a large hydrophobic region which can partially insert into the membrane, and so that's going to disrupt the membrane and be surrounded by the fatty acid tails or the phospholipids. Then we have transmembrane proteins which have single passes, and so this molecule here is glycophorin A. It's a dimer of glycophorin A, so there's one N terminus there, and if we trace it all the way around, we come to a C terminus down here. So when we have proteins which are integral membrane proteins with a transmembrane helix, just one of those is what we see is the N terminus on one side and the C terminus on the other side. And so that's because the transmembrane helix passes once. And so there's another molecule here to the right with a hydrophobic transmembrane helix, and these associate to form what's called a coiled coil. The hydrophilic faces of the two coils wrap onto one another, and they kind of have a complementary face that fits together, and you get this dimer of this protein. It's a nice example of quaternary structure in a membrane protein there. Now these chains have to be wide enough, so these helices have to be wide enough to get across the membrane. So if the membrane becomes wider, then you have to have more transmembrane helix. Now you can't just suddenly make transmembrane helix, that's a consequence of the amino acid sequence. And so you will often see certain proteins being limited to certain membrane compartments or lipid rafts based on the width of the membrane they can span with their transmembrane helix. Other transmembrane proteins fold into what's called a beta barrel configuration, and so here we can see that these beta sheets are wrapping around in here an anti-parallel conformation, and this creates a channel through the middle. And so this is common in porine proteins like the aquaporins, through which water can pass down a free water concentration gradient. And so that's another configuration. That's called the beta barrel. Particularly beautiful example of protein structure there, the beta barrel. When we look at multipass membranes and multipass transmembrane proteins, these are proteins which are going to have helices which go back and forth, and so you may end up with the N and the C terminus on the same side of the membrane, or the N on one side and the C on the other, because these membrane regions go back and forth across, and these are often, if not always, alpha helical in shape. So here's bacteriorhodopsin. This is a protein which certain bacteria use to capture light energy for photosynthesis, and there is a prosthetic group, retinol, which can be used to capture light energy. Another example here of a multipass transmembrane protein, glycerol channels through which glycerol can pass in and out of cells, and there you can see the helices are arranged slightly on a slant, and they're going to be kind of in a barrel, not unlike the beta barrel, but here the defining secondary structure feature is the alpha helix. These transmembrane regions can be anywhere from 2 segments to 20 segments. In some cases you may even find examples with more than that. So there's a nice example of some multipass transmembrane proteins. Let's finish up looking at lipid anchored proteins. So the polypeptide component of a lipid-anchored membrane protein is located on the surface of the membrane, either the outer leaflet surface or the inner leaflet surface. They're covalently bound to some kind of lipid molecule, a myristal, farnesyl, palmitol, GPI, that kind of thing. So the protein is actually attached to a lipid-like or a lipid structure. And as you can see here, there are any number of types of lipid or lipid-like structure that can be used. Here's a myristal anchor. So that's a saturated hydrocarbon chain that's been attached to the protein via an amide linkage. And that myristic acid then attaches this completely cytoplasmic or extracellular protein to the cell membrane. So there is a wide variety of these types of anchors. Their characteristic is what's important though. They're all going to be hydrophobic, so they can embed into the membrane, much like a fatty acid tail would embed into the membrane. So that's it, a little bit about membrane proteins. This is the huge section. A lot of it is review in your text, and a lot of it is review with a little bit more information that I think you can handle really easily on your own. So I'll wrap it up there. I'll see you next time.